The overall goal of this procedure is to image fluorescent T cells in lymph node slices by Widefield and Confocal microscopy. This is accomplished first by removing a mouse lymph node and embedding it in low melting point agarose. The node is then cut into 320 micron thick sections with a vibratome. Next, fluorescently dyed T cells are plated on top of a lymph node slice. Finally, the T cells that have infiltrated the slice are visualized by Widefield and Confocal microscopy. Ultimately, still and videographic images can be obtained that show how T cells are recruited into the lymph node and what kind of motile behavior they exhibit within the lymph tissue.
Okay, the main advantage of these techniques over existing methods like two-photon microscopy on intact organs is that here the T cells can be visualized in a three-dimensional system with a conventional wide field microscope. And moreover, the slice preparations permits an access to the tissue.
Begin by preparing a 4% low melting point agarose solution by diluting agarose in PBS and microwaving the resulting admixture. After the agarose has completely dissolved, maintain the solution at 37 degrees Celsius until use. Next, fill a plastic dish with RPMI complete culture media and place stainless steel washers with inner diameters of 4 mm into the dish. Fill another dish with ice-cold PBS. Then add approximately 1.1 milliliter of RPMI complete culture media per well in a 6-well tissue culture plate.